Good morning, everyone. Today we are going to start with the rodent memory testing playbook. Okay, so this slide shows the taxonomy. Of the behavioral memory essay. The rodent memory essay can be broadly classified according to the type of cognitive memory domain they assess. The first category is the recognition memory, which evaluates the ability of a rodent to discriminate between the familiar and novel stimuli. The novel object recognition test is the classic example. The second category is a working memory, involves the short-term storage and manipulation of the information required to ongoing tasks. The Y-maze and T-maze paradigms are most commonly used for this purpose. The third is spatial reference memory, which depends heavily on the hippocampal function. The Morris Water maze and Barnes maze are considered the red gold standard essay in this domain. Finally, the associative learning paradigm evaluates the ability to form association between the stimuli and outcome. These are commonly assessed during using fear conditioning models. Sections of the appropriate test depends on the target brain regions, disease model, age of animals, stress sensitivity, motor preference, performance, duration of memory being tested. No single assay can measure all the aspect of cognition, which is why multiple paradigms are often combined in experimental neurosciences. Now, this slide shows the novel object recognition test. It is one of the simplest and most commonly used behavioral paradigm for evaluation of test for evaluation of recognition memory. Okay, involving the short term, involving the short term and long term memory. Okay, so into this for this. Okay, okay. The first category is okay. Into this, the scientific basis of this test relies on the innate exploratory behavior of mice, a rat, depending which spends more time in investigating a novel object than a familiar one. The test generally consider of a four-phase habituation phase. The rodent is allowed to explore an empty arena without subject. Okay, for 15 to 30 minutes. Then familiarization or encoding phase. Two identical objects are placed in the arena. The rodent explore both objects, creating a memory trace. Retention interval. The animal is removed from the arena for a specific period. Depending on the duration, researchers can evaluate the short-term memory or long-term memory. Okay, long-term memory retention interval. The animal is removed from the arena for a specified period. Depending on the duration, researchers can evaluate the short-term or long-term memory. Now, the last one is the test phase. One familiar object is replaced with a novel object. Increased exploration of the novel object indicates the successful memory retention. The primary outcome measure is the discrimination index or a preference index. The advantage of this test include the simplicity, non-invasive methodology, no requirement for the reinforcement or punishment, minimal stress to the animal. However, the test is highly sensitive to the anxiety, locomotor activity, object preference bias, lighting, and environmental condition. Therefore, the proper experimental standardization is required. Next, the Y-based test. Okay, this test is used to evaluation of spatial working memory and exploratory behavior. The apparatus consists of the three identical arms arranged at 120-degree angles, forming a Y-shaped structure. Rodents naturally prefer to explore unfamiliar environment. Therefore, they tend to enter a new arm rather than returning to one they previously visited. This behavior is known as a spontaneous alteration. During the experiment, the sequence of arm entries is recorded over a fixed duration over 5 to 8 minutes. The percentage Alteration is calculated using the formula actual alternation divided by the maximum possible alternation multiplied by 100. High alteration behavior indicates intact memory and executive function. The assay is used in Alzheimer's disease, schizophrenia, aging research, pharmacological screening. Advantage of the YMAs include rapid testing, no requirement for the food preservation, low stress environment, easy experimental setup. Limitation include the dependence on the locomotor activity, sensitivity to anxiety, potential confounding by the hyperactivity, sedation. Therefore, locomotor control measures are often incorporated into the study design. Next is Morris Water Maze test. This is considered as one of the most important and validated assay for the assessing the spatial learning and the reference memory. The apparatus consists of the large circular filled with the opaque water. A hidden escape platform is submerged below the water surface. Rodents are trained to locate the hidden platform using the visual clues placed around the testing room. The essay usually includes acquisition or learning trials, probe trials, reversing learning in some studies during acquisition phase. Acquisition trial, the animal learned the platform location across the repeated session. Important parameters include the escape latency, path length, swimming speed, search strategy. In the probe trial, the platform is removed. Memory retention is assessed by measuring the time spent in the target quadrant, number of crossing over the former platform location. The Morris water maze is highly dependent on the hippocampal integrity and it is extensively used in the Alzheimer's disease research, stroke model, neuropharmacology, aging diseases. Advantage: strong translational relevance, high sensitivity to the hippocampal dysfunction, quantitative data acquisition. Limitation is stress induced by the swimming, requirement for the intact visual and the motor function. Possible hypothermia is prolonged testing. Proper animal welfare consideration are therefore extremely important. Now, the next one is Barnes maze, which is a dry alternative for the spatial learning. This consists of the circular elevated platform containing multiple illuminating open spaces into the dark enclosed area. Okay, and rodents are naturally motivated to escape from the bright illuminating open spaces into the dark closed area. Animals learn to locate the escape box using the external spatial clues. Key outcomes measures include escape latency, number of errors, path efficiency, search strategies. The Barnes maze is particularly advantageous in aged rodent anxiety sensitive model studies where the swimming stress may confound results compared to the Morris water maze. The paradigm causes less physical stress and avoids complication related to the water exposure. However, motivation might be weaker than in a water-based task, and animals might adopt the random search strategy rather than a spatial learning. Despite the limitation, barn maze remains a valuable tool for evaluating the hippocampal dependent spatial recognition. Now, the slide shows the right as the diagnostic matrix. Okay, oh, as the researchers must consider several factors before finalizing the experimental paradigm. First, the cognitive domain of interest must be identified. Recognition memory, like the which memory you are going to study, like long term and objective memory, long term, short term spatial working memory, spatial reference memory, and spatial learning. Okay. Second, the disease model must be considered. For example, Alzheimer's disease studied often involves Morris water maze test and NOR, like that. The physical and sensory ability. Okay, like primary successful. Okay, like driving motivation. What are the driving, like we know that in novel object recognition test we can study the objective like short term and long term memory, and the driving force motivation is the innate curiosity, and the apparatus is open field, and we will study on the basis on the novel versus familiar time. Familiar time. Okay, why base involves the study of why may involve the study of spatial working, short term memory, and in which the driving motivation is the innate curiosity. Apparatus is y shaped arm. Primary success matrix is spontaneous alteration. The test name next one is the Morris Water maze, in which we study the spatial references, and driving force is aversion, and apparatus is epac pull. Target and the primary successful matrix is targeted quadrant tribe. Barnes maze, spatial learning, aversive light and open, and apparatus is elevated platform, and the next one is the latency phase is latency and error count. Okay, now the foundation of validity and critical laboratory based practice. Handling animal for 1 to 2 minutes daily for 1 to 2 week prior to testing impact dramatically reduces the baseline stress, preventing skewed exploratory behavior. Environment: maintain the low diffuse light, keep the testing room acoustic isolated and strictly consistent. Odor control: clean the testing apparatus thoroughly between every single trial using the 70% alcohol. Impact: eliminates the residual olfactory cues left by the previous animal. Objective tracking: utilize the automated video tracking software like enemies. Impact: remove the human observation bias and ensures the precise temporal and the spatial data collection. Now the conclusion. Okay, so in conclusion, rodent memory cannot be directly observed. It must be carefully extracted through the spatial and objective based proxies by matching the right memory domain to the appropriate biological motivation and adhering strictly to the handling and tracking protocols. Researcher can capture objective, reproducible data from this experiments. Okay, thank you for listening.